I know this has been a long wait for a lot of you that have been waiting patiently for COVID-19 conspiracy question mark part 6, empowering yourself. I hope you enjoy this video. We did have to break it up into 4 parts because one, it's kind of long, and also we weren't sure if YouTube and Facebook would censor this. You'll find that there's not much in here that should be censored, but then of course, even our message to the world about holding a place in your heart with love, gratitude, compassion, and asking source energy to help support this evolutionary period for humanity. That message that Dr. Lipton, myself, and Nia Peoples had sent out was censored. So we don't know if it'll be censored, but suffice it to say that the entire video is available on the Advanced Medicine dashboard, where you have access to not just this video, but you'll have access to all 6 parts and the 7th part that'll be coming out right after part 6 is completed. And you will be able to download each one of those videos. I want you to spread those videos far and wide, post them wherever you want, keep them on your devices, share them with friends and family, because it's about time that the world recognizes what the truth is. And you having those videos in your hands, in your possession, being able to disseminate them easily is the only way that that's going to happen. So pay it forward, pass it on. Thank you.
All right. All right.
So go ahead, Bruce. What was the question you were asked Judy?
I was asking my expert, because I'm giving a lecture on it, and we talk about the HIV and the GP120 and all that kind of stuff like that. And from the brief discussion I heard from you, you actually come to the conclusion that it was a cytokine cascade that was the problem, not the loss of the CD4 cells.
Correct. So what my PhD thesis said was that, at the time in the mid 80s when I started my work with Dr. Frank Rossetti, it was 1987. And basically, because Fauci, and we didn't know this until recently, because he had the grant, the patents on IL-2 therapy, which you'll recall Frank Rossetti discovered. So it was a big surprise to us he had the patents. But for that entire decade since Luc Montagnier isolated the virus, and we confirmed the isolation with the same patient population, what we've been investigating is how the virus caused cancer caused AIDS, when in fact only 1 in 10,000 T cells was infected. So if only 1 in 10,000 T cells is infected, of course, the other, the hypothesis is somebody else is the orchestrator of the cytokine storm. And so that work, we proposed the monocyte macrophage, infected primary monocyte macrophages, isolated primary monocyte macrophages away from HIV infected people, showed what kinds of energies, what kind of toxins, what kind of co-infections activated the latent expression, expanded the viral reservoirs, and turned the fire up on the storm. At the same time, as we identified the NF-kappa B pathway, it was prior to, so the NF-kappa B pathway and that transcription factor created the IL-6, the IL-8, the storm of the flame of the signature. At that time, NF-kappa B was thought to be totally a cytoplasmic protein, nuclear factor kappa beta, and regulating B cell and antibody expression. It wasn't really known how it functioned. So we submitted the paper where I isolated the nucleus after my PhD thesis. My PhD thesis was, is the macrophage infected and the orchestrator of the immune response? They told me, oh, you're ridiculous. Macrophages don't have CD4 on them. And this was, of course, before we understood the delta CCR5, the delta 32 deletion in people that had HIV and would never get AIDS, because it was that CCR5, that chemokine receptor 5, which was the target of peptide T. So we knew peptide T worked back in the 80s, and we didn't know why. So that's an immune therapy, an immune modulating agent. And all we need do is keep that CCR5 T cell interaction from happening, and you would never get AIDS. And so with because the virus couldn't enter through CCR5. So the first publications we tried before my PhD thesis is you're an idiot because macrophages don't have CD4 on them. And your macrophage cultures must be contaminated with T cells, which of course wasn't what turned out to be. And I did defend that thesis and show what activated it from latency. So there were really two things: DNA methylation. We did this work with Stephen Balin in my postdoctoral career to continue to ask what happened. And so what we knew is that the retroviruses have CpG on-off switches in their promoters, and so do immune response genes. And so the question that Steve Balin asked us originally in 1992 was, does DNA methylation silence retroviruses and prevent them from doing harm? Which to me was quite boring, and to Frank Rossetti was quite boring, because yes, that's certainly true, and there's no reason to expect, and we had in vitro data, so that's boring. So what we asked was, what do retroviral infections, what does HIV do to the DNA methylation machinery? And that's really the key. And it's the key to, and so what it does is it basically hijacks the DNA methylation machinery and alters the methylation patterns. And it gives you a broader range of epigenetic silencing through DNA methylation. And it silences tumor suppressor genes, immune response genes. And in the case of the study we did, what we did was we looked at the promoter of interleukin-2 and interferon gamma, Th1 responses to get a protective immunity against the virus. And they only have one CpG different in the promoters of interleukin-2 and interferon gamma. So what that says is, and we took advantage of a restriction endonuclease known as SNAB1, which is it's methyl sensitive. If it's methylated, SNAB1 won't cut that site, and if it's not methylated, it will. So the DNA methylation, as you know, is the stop sign on the promoter. Don't transcribe that gene. Don't write that virus. Don't express that virus. So did a simple PCR assay across that, with and without SNAB1. And very key, the reason why HIV/AIDS patients don't have a type 1 immune response, it's crippled and they can't silence the flame, and is that single methylation and the alteration of the methylation machinery in HIV infected individuals. So it's two things. It's the inflammatory signature, and F-kappa B translocates to the nucleus. We didn't know about I-kappa B. We published that paper. They said, you're an idiot, your cells are, we threw it in the desk drawer because Frank Rossetti never cared about publications. And on the minute they cloned I-kappa B and recognized the phosphorylation releases to the nucleus, then in fact the signatures turned on. Well, nothing turned the signature off because the virus had at the same time hijacked the type 1 interferon, the off cells. And it's very important with respect to COVID-19, a paper that just came out with TGF-beta, Frank's other expertise as a regulator that, let's just call it a master regulator, bifunctional regulator of the hematopoietic stem cell. Well, what that paper says is that TGF-beta through STAT3 causes a hypermethylation and silencing of SOX3, which is suppressor of cytokine signaling 3, which we didn't know about in the 80s. So now SOX3 is suppressed. So you can't suppress the inflammation driven by SARS-CoV-2. At least at the level of the lung and TGF-beta, drives fibrosis in people who already have that trigger switch. So the idea that you have resident stem cells now, tissue resident stem cells of macrophages throughout the body, and it's not the bone marrow macrophage, because again, we didn't know there were resident macrophages in each thing in 1987 or 91. So we can start putting all of these things together. And with respect to HIV, yes, the infection alone, we did further work on that DNA methylation alone to show you didn't need infectious virus. You don't need GP120. You don't need the envelope. All you need is RNA or DNA to stop the methylation, to alter the methylation machinery, and that sets the cascade going. So it's really key in this case, where now we're injecting, and Luc Montagnier has been tapping us the last few weeks, and he and Frank Bracetti over the last few weeks. Frank and I still work together in Carlsbad, California. And he's been tapping us about the HIV sequences in this new strain of virus. And so we see an ability with some strains, just GP120, which doesn't make sense to me, but in his original work, he was showing us GP41, which is, of course, the transmembrane. So if you just have GP120 and you don't stick to the cell, you're floating around, that's not a big deal, in my opinion. So there's something wrong with that. So at any rate, what we see with SARS-CoV-2 is an expanded host range, because, you've got the spike protein and you infect the epithelial cells, fine. But why are we seeing coagulopathies? Why are we seeing other things that look like HIV or retrovirus, psychoses, and things like that? Coronaviruses don't infect the brain.
Well, they don't, unless you've taken them through a chimeric process and manipulated them and altered them and adjusted them from their natural wild type morphological status. So
correct, yeah. Which is clearly why Luke agrees with us in all of this,
right.
All of this stuff. So does that more or less answer the question?
I guess because when I'm teaching, I talk about 5 immunopathologies, and immunodeficiency, of course, is one. But now it seems like I should put HIV into a control mechanism dysfunction and not an immune deficiency. Does that make sense?
Yeah, in both it fits. So
it fits in both categories.
In both categories, almost. And I do something, and I can share with Rashid the PowerPoints and our papers that matter with respect to the XMRVs and the HIVs as well, because it's really key in all of this. And even a couple of papers that I think I've already shared with him, but I'll do it again. On just the whole idea of naked DNA, naked RNA, protein, the amount of danger-associated molecular patterns that we're injecting, and altering DNA methylation machinery, because you use the substrate. You pull the substrate off your islands in your in the rest of your genome. So all the knots on a string the express genes are heavily methylated, and we have huge islands, CPG islands, and they're heavily methylated. But what's happening, because of all we're injecting that are naked CPGs, that's a danger associated molecular pattern. So we're crippling the immune system, and it drives the disease engine, reactive oxygen, nitrogen, the cytokine storm, everything that hits you, and that's how HIV can be driving this, because we now appreciate in many, many, many of the cases of HIV, the infection wasn't from without, it's from within.
I appreciate your insight, because it's going to redirect my lecture. And so I really want to be on board with the right story for today. So thank you for spending your time to let me get more information on that. Thank you.
Yeah. You're welcome. No worries.
So what I'd like to do is we're going to have two different audiences here. And I think it's important that we make sure that we cater to both of them. One is going to be the professional, medical, scientific community. And I don't know whether you guys think that we should just talk first on a superficial level and excuse me, I don't mean superficial level, but we talk more on a fundamental level that the layperson will understand, and then we go more into the scientific aspect, or whether we should just let it loose and then let everybody see it. I don't know how you guys feel about it, but part of me feels like the public is so much more educated, and we want to empower them with as much information as possible, so that we should probably go and just have just one version and let them we should always keep it as simple as we can in the beginning, and then we can expound upon it in more scientific detail if we need to. Is that good with you guys?
That's great.
Perfect.
It's great for me. I've taught in medical school for years. And when I started talking about the biology of belief, I had to really say, look, I have to start from the basic beginning here, because your education has distorted everything. And so we need to start over here. So I had to talk down, actually bring it from the bottom up for them to get it. Because if I started up here, their confused information down here will distort everything I said up here. So we had to start from the beginning.
Right.
So I really feel we address this to the conventional audience. And let medical doctors pick up what they can from this, because if we jump at the top, I think they're going to distort everything we said in the first place.
I agree. And plus, I was trained in general surgery, so we were very simple-minded people. When in doubt, cut it out and heal with steel. So we need to keep it very, very fundamental of ABCs, and that's it. Yeah. Because beyond that, I may not get it. So, all right. So I'd like to start off first by talking a little bit. I have a philosophy about the 7 toxicities. And in my experience, in my clinical experience, when I discovered your work, Bruce, 16, 17 years ago, it was actually 3 toxicities at that time. And as time went on, these toxicities, they're not actually in the order of significance. They're more in the order of how they were revealed to me, or how I discovered them, or how I experienced them. So I'm going to first quickly go through those 7 toxicities and talk about the current situation, how they relate to each of these toxicities. And then I would like to use that to expound upon. So in the 7 toxicity philosophy, basically the common theme in when dealing with these 7 toxicities is that all these toxicities lead to one common etiological component that causes all disease, all pathology, and that's oxidative stress. The first one is heavy metals. So before I go into the individual ones, my belief, my postulate 22 years, 23 years ago was that if this is true, that if it's oxidative stress where all injury starts from, then by definition, if you can eliminate oxidative stress, then you should either reduce pathology, but potentially, theoretically, eliminate it completely. So that's the postulate I began with. And now I can say 2 decades later that without any doubt, that if you can effectively detoxify the body of these 7 toxicities, and the key operative word here is effectively, if you can effectively detoxify the body of these 7 toxicities, by definition, chronic disease cannot exist. Cancer, heart disease, diabetes, arthritis, whatever it is, they cannot exist. So what's the first toxicity? Heavy metals. Now, this is not just the heavy metals like mercury, nickel, and all those types of things, uranium, plutonium, etc., but it's also the trace minerals in higher amounts than they should be. For example, iron, selenium, copper in higher doses than what the body needs actually start acting like heavy metals. So that's the first toxicity. The second one is the persistent organic pollutants. The persistent organic pollutants, also known as POPs, include the organophosphates, the fluorinated hydrocarbons, the insecticides, pesticides, all the different chemicals, etc., etc. Now, when you start looking at those first 2 toxicities, looking at the current pandemic state, or what they're calling the pandemic state, there is a very significant foundation that has been put in the global society to increase the level of toxicity, from chemtrails to where everything started in Wuhan. It was all the outgassing from the incinerator burn-off, and it had been so significant that it garnered CNN and BBC in 2016, 2017 to cover the level of toxicity that the people in Wuhan were dealing with. And it's interesting that in the COVID-19 conspiracy question mark videos that I put out, The first one was facts versus fiction. The second one was toxicity ignored. So when we go into this component, now we're looking at the toxicity that has been impregnated into the society, into the global society, almost like a setup, almost like a foundation, like plowing the field and preparing the body to be more susceptible to what's coming. The third toxicity is opportunistics, the bacteria, viruses, spirochetes, mycoplasma, yeast, parasites, et cetera, et cetera. I don't think we need to say much about that. That's where the COVID-19 fits into that picture. The fourth toxicity is the energetics, ambient cell phone radiation, electromagnetic radiation, radio frequency, electromagnetic fields that are being created, et cetera, et cetera. And that brings in the whole topic of 5G, which is amazing to me. Yeah, I just said it, right? So we're going to have to make sure that we maybe highlight it or bleep it out or something. But what's interesting to me is that if the 2,000 studies that have been put out just regarding that one component weren't relevant, then why is it such a huge issue that all the major channels, Facebook, YouTube, Google, everybody's censoring it? I mean, if it really was a conspiracy theory, and it's funny how we use the word conspiracy now, right? Conspiracy theory means something that is conspiratorial, and that's exactly what this is. But they're trying to, it's almost like how mainstream medicine uses evidence-based. They do everything evidence-based, and it's anything but evidence-based, so they try to hijack the terminology and try to take advantage of it, our terminology. So anyway, that's the fourth one, energetics. Fifth one is the emotional psychological one. Now, if there was one that was more important than any of them, it would be the fifth one. We know this, how important this is in cancer. It's a belief system. I know, Bruce, you are the expert. I learned everything about that aspect from you. I'd say 90% of it. The rest of it was from people like, Andrew Carnegie, Napoleon Hill, and Maxwell Maltz. But you really put it into picture, into frame of reference from a medical scientific standpoint. But what I find interesting now is currently what's going on with this COVID-19. They are really hitting that one the most significantly out of all the 7 toxicities with the fear aspect, the 6 feet social isolation, masks on your face, staying home, blah, blah, blah. The sixth one is foods, not what we're eating, but what we do to our foods, the homogenization, pasteurization, irradiation, genetic modification. So how is that applicable to the current situation? Well, they've been prepping us on that as well with all the genetically modified foods, the glyphosates in our food system, and over the years. So toxicity 1, heavy metals, toxicity 2, persistent organic pollutants, and toxicity 6, what we do to our foods, have been something that's a long-range plan, I believe. And then the last one is spiritual toxicity. And that's the one that I think that they're trying to affect us. And Bruce, I'm happy to tell you, we've got over 100,000 views. It was released this morning, 8 o'clock, the video that you and I and Nia did. Other people have made their own versions of it and put it out there. And so it's just starting. And this is what I wanted. I want to have some momentum before we get into the Saturday night, Sunday time frame, which is the most critical time frame. So this is the 7 toxicities that we're talking about. They've hit 6 out of the 7, trying to take the general mindset and population, the global population, making them weaker, making them more susceptible. And so I'd like to use that as a theme as we go forward to start talking about how we can impart knowledge and empower the individual, each separate individual out there, no matter where they are on the planet, and help them to negate some of these things that have been orchestrated to try to prevent us from staying healthy. So from that standpoint, from a clinical standpoint, obviously exercise, obviously eating healthy, obviously getting rest, adequate hydration, washing your hands. My kids say, Dad, what's the difference between this and what we've been doing for 20 years? We don't wear shoes in the house, basic things like that. So that's something that people should know. I'm going to talk about more of the clinical aspect after Judy. You and Bruce, you go into more details from your perspective. So Judy, let's start from the mask aspect, if that's okay. Let's just talk about some specific things that we're being told to do right now and how detrimental they are, actually, how counterproductive and contrary they are to maintaining health. So can we start with that?
So this is an Anustat is a natural product drug that we've been developing in consultation with somebody since 2005. So forget the title, but in cancer, but I told her to change everything to COVID. So what I heard you just say in all of those things you said, the microbial switch and the various triggers, right? There is ROS and reactive nitrogen species. There's a typo there. But that's the idea. You create this disease engine, starting with oxidative stress, and then microbial infection if it is an infection, but also injection, and then leading to inflammation. And chronic inflammation is, of course, the killer as you drive the tissue pathogenesis of the host. So to get to the mask, where that matters, number one, and I know Bruce will address the mask from the standpoint of the fear, but from the immune suppressive parts that you just talked about, that he just talked about, that you're actually activating your endogenous microbes. So you're activating the expression of other RNA viruses, other microbes that in fact impart themselves on and on the disease engine, and cause the problem in the disease engine and drive that. And I'm also going to show that from the standpoint of another paper I tend to carry around with me. Let's see if I can find it.
Judy, while you're finding that paper, let me just ask you this, and actually it's a rhetorical question, but I'm going to ask it because it's, the fundamental aspect. We are beings that need oxygen, and
Correct.
Plants are beings that use carbon dioxide. And if we are
Correct.
restraining with the airway restriction, then of course we're getting less oxygen. We're actually stressing our system because we now have to suck the oxygen out of the air through this filter. And we're creating all this other stressors, the psychological stress of oh, I got to have my mask. And so anyway, I just wanted to kind of open that can of worms up a little bit.
And that's exactly what I'm saying. So all of those stressors are actually extremely immune suppressive, and we're not, as you said, meant to breathe carbon, our own dirty carbon dioxide waste. We're intended to breathe oxygen. So as we've seen, so this is the paper that I'm showing you now. So this paper is called War and Peace Between Microbes, and it's from 2010, I believe, by our colleagues in the National Cancer Institute in drug development, and the first author is Andrea Lisco there. And so what that shows you is how, in this picture here, and it's with HIV. So here are normally symbiotic viruses in the cell. This is figure 1 from that paper. And as you said, when you stress the system, you activate, you've got oxidative stress because you're not inhaling pure air. That by definition is oxidative stress number 1 in your injury thing. And so then you stimulate the expression of what would be considered symbiotic viruses. And what they do, those reactivation of symbiotic, you're in equilibrium with them. They're not a problem to you. Your immune system has them in order. And what that does is it will, that you see them in balance in figure A, with triggering immune responses and containment of viral replication. You keep the virus silenced no matter what kind of virus it is, whether it be EBV, herpes viruses, and things like that. And then in figure 2, you can see with respect to HIV, and for your class, I'll make sure you get this, Bruce, you can see the downregulation of antigen presentation, the downregulation, the abnormal production of chemokines, the transactivation by transcription factors, as we just discussed, making more and more, HTLV1, HSV2, HHV6, things that we know have been associated with these chronic AIDS-like immune deficiencies from the disease engine. So the mask is just one of the worst things you can do, because we all have those symbiotic viruses. So we immune suppress through, 5th and 7th of your toxicities. And then we create that disease engine, because that microbial switch doesn't have to come from without, it comes from within. And so then what you're activating is your own coronaviruses that you may have silenced, and you're creating recombination between pieces and parts, and you're just crippling your immune system, taking out your type 1 interferon, your basic antiviral response, because you're and natural killer cells, their only job is to detect cancer cells and virus infected cells. And they do that through stop signs, killer inhibitory receptor. Don't eat me. It's a Pac-Man. Don't eat me. So we're suppressing our natural, at various levels, our natural immune system.
In fact,
just by wearing that abnormal mask.
In fact, it's the whole lymphocyte subpopulation that we start to decrease, is it not? Because you're creating.
As that talks back and forth.
Exactly.
Absolutely.
The entire.
The regulatory cells, the myeloid suppressor cells, we create just, a nightmare.
And now that you brought that up, I feel like this is a basketball team, like a perfectly tuned basketball team, because I think that I threw you the ball and now you've alley-ooped it, and now Bruce is going to slam it through the hoop, because now that brings up the whole aspect of exosomes, because you're talking about these things that are within ourselves. So now, Bruce, how would you say, when you look at the exosome theory and then you look at the viral theory and you look at Steiner's work, and Steiner showed that it's actually the release of viral load. It's secondary to a toxic epigenetic environmental trigger that causes this release. And then you have, there was a doctor, I think his name is Kaufman in the West Coast, that put out a video that was a really well done video where he shows the similarities between exosomes and viruses, specifically the COVID-19. And there's no distinction. I mean, there's, morphologically, characteristic, size, electron microscopy, everything. So would you like to take that one, Bruce? Would you like to slam that one through the hoop? I hope you enjoyed this first part. Part 2 will be released tomorrow. Tune in, download it, spread the word.